In this section, we'll continue to look at different binding interactions that can occur between a drug molecule and its intended target. So in this section, we're going to discuss van der Waals interactions, dipole-dipole interactions, and ion-dipole interactions. Now we're going to look at van der Waals interactions. Now these are some of the weakest interactions that you can get with a strength of 2 to 4 kilojoules per mole, but the sum of these forces can add up to a significant effect, and so they are still very important in the context of drug-target interactions. Now these occur most in hydrophobic regions, especially in regions containing lots of carbon-carbon and carbon-hydrogen bonds. So what is a van der Waals interaction? Well, I want you to imagine that you have a molecule. So you have a molecule here. This molecule is contains no charge, and it is, for all intents and purposes, neutral. Now in this molecule, you can have slight movement of the electrons. So these are natural movements of the electrons backwards and forwards as they go around the nucleus. And what happens is that as these electrons change position, you get a very, very slight positive charge on one side of the molecule or of the group, and a very, very slight negative charge on the other. And that this effect goes backwards and forwards so that you are alternating which side you have your positive charge and which side you have your negative. Now this is called a transient dipole. Now overall, on average, the molecule is neutral, but you have this very, very slight difference in charge between one side and the other, as the electrons move slightly backwards and forwards. Now this transient dipole, when the group containing this dipole approaches another neutral molecule, can have an effect on this molecule to cause an interaction. Now what happens is the dipole on the original molecule affects the electron distribution in the second molecule. So for example, if the original molecule has a slight positive charge here, it will slightly attract the electrons in the functional group on the second molecule. And so it will very, very slightly attract the electrons in the second molecule towards the original dipole with the slight positive charge. So we end up with a slight positive charge on the original dipole and a slight negative on the other. And this is an attractive force which attracts the groups together. Now these two dipoles will still be resonating backwards and forwards, but they'll do so in sync and still have an attractive interaction. So if we're going backwards and forwards between, for example, the positive being on the right hand side and the negative being on the left, and then the positive being on the left and the negative being on the right, the opposite will occur in the second molecule. So we'll have the positive on the left next to the negative charge, and then the negative charge will be on the other side of the molecule away from the positive. Now this interaction is an attractive one, and it's one that can help hold these functional groups together. Now in a drug binding site, the same thing can happen. So here we have our drug molecule, and in light blue we have our hydrophobic region. Now when this approaches a hydrophobic region in the binding site, which is shown in dark grey, we can get this transient dipole induced. And this transient dipole forms the basis of an attractive interaction between the drug molecule and the binding site. Now this is called a van der Waals interaction. The van der Waals forces are the forces that dictate the movement of the electrons and the subsequent attraction between the groups. And so this interaction can form between the drug and the binding site on the target. Now this interaction is very weak and it's not felt over any significant distance. So unlike an electrostatic interaction, this does not help the drug get into the binding site. But once the drug is there, this interaction can help hold the drug in the binding site and secure it in place. So we just had a look at van der Waals interactions, which involve the formation of a transient dipole. But what happens if we have a permanent dipole on a functional group? Well, if we have a permanent dipole, we can get a dipole-dipole interaction. So we get one dipole interact with another. The presence of a permanent dipole means this is, relative to van der Waals interactions, a much stronger interaction. Now, the polarization of a bond by a presence of an electronegative atom produces this permanent dipole. So think of functional groups like a carbonyl, for example. And you can get a positive interaction between the dipole in the drug molecule and the dipole in the target site. So what does this look like? Well, here we have a carbonyl group. This possesses what's called a dipole moment. So we can represent the dipole using an arrow that looks like this. So the cross on the arrow represents where the slight positive charge is within that dipole. So the slight positive charge in a carbonyl is on the carbon atom. The arrow head represents where the slight negative charge is. So in this case, it's on the oxygen within the carbonyl. So we can say that this dipole has a directionality to it. And dipoles will line up with a dipole that is going in the opposite direction. So you can imagine that the positive charge in this carbon would be attracted to the slight negative charge of another carbonyl. And so if we have a dipole moment on our molecule, or on our functional group, this can line up with a dipole moment in the binding site. And so we would get the functional group approach the binding site in the opposite direction to the dipole within that site. So as I've said, dipoles go from a positive charge to a negative charge, and so that will align in the opposite direction to the dipole within the site, because you'll get the negative charge attracted to the positive and the negative attracted to the positive. So this type of interaction not only helps hold the drug molecule in place, but it also helps get the drug molecule into the correct orientation. Now again, this type of interaction is not felt over significant distance, so unlike electrostatic interactions, this can't be used as an initial driving force to get the drug molecule into the binding site. So we've just seen what happens when you have transient dipoles induced, but what if you have permanent dipoles on groups such as carbonyls, for example? Well, permanent dipole moments also have an associated direction, and so correct affinity of these dipoles will result in much stronger interactions than when using van der Waals forces. And they can be used to help the drug bind into its binding site. So the inclusion of these dipoles, permanent dipoles, can increase the strength of which the drug binds to a binding site. But it's not just carbonyls or individual functional groups that can have a dipole. Whole molecules can have a dipole that is spread across them. So one part of the molecule could overall have a slight positive charge and the other one have a slight negative. And if a molecule has an overall dipole, we can again use this to correctly orient the molecule within a binding site. So let's have a look at some more complex molecules and their associated dipoles. So here we have cimetidine. Now this contains a large variety of heteroatoms, so it contains nitrogen atoms in many different functional groups, and we also have a sulfur atom as well. Now if we look at a couple of variants of this molecule, we have this one here, which contains a nitroketonomal functional group, which is here, and this nitro group in particular is very electronegative. And we also have this derivative here, which contains a imidazolinone group, which you can see here. Now if we have a look at the dipoles along each of these groups, we can see that the dipoles can go in different directions. Now we represent the dipole in the same way as we would with a single functional group containing a dipole, in that again we have a positively charged base, the dipole, and a negatively charged head. And each of these groups has a different dipole orientation associated with it. So for example, here is the dipole from the cimetidine, and it has an angle of 13 degrees. So this dipole is going 13 degrees relative to this carbon-nitrogen bond. Now, as we move across these groups, we can see that the directionality of the dipole changes. Now, the term below this is an indication of the strength of the magnetic dipole going across each of these groups. And you can see that depending on the group, this strength changes. Now, you do not need to memorize any of these groups, and you do not need to remember any of the angles associated with their dipoles. But you should be aware that permanent dipoles can be associated with a functional group or an entire molecule, and that they can help to correctly orient the molecule within a binding site, and they can help form relatively strong interactions that will increase the binding affinity of the drug molecule. And just to give you an example of how this can help orient the molecule in the binding site, here we have our molecule with a permanent dipole moment. So remember that the dipole moment goes from positive, with the end of the arrow that looks like a plus, to negative, where the arrowhead is. Now, on our binding site, on a receptor, we could have a binding site that contains hydrogen bonding sites, so these are represented by these blue circles, and also contains a permanent dipole, so containing a positive charge at this end and a negative charge at this end. Now, as the drug molecule approaches the surface, the dipole could help orient this molecule in the right position. So if we have placed hydrogen atoms in the correct orientation in the molecule, in the correct position, not only could we have a dipole-dipole interaction, but we could also facilitate two hydrogen bonding interactions as well, giving this drug a very secure binding site on the surface of the receptor. However, if we design our drug molecule incorrectly, what we could get is the dipole in the molecule, again orienting the molecule in the binding site, but this time the hydrogen atoms could miss the hydrogen bonding sites within the receptor. And as they miss these sites, it results in a much weaker interaction between the drug and the receptor. And so if we know what dipole we need to target the surface of a receptor, we can try and design our drug molecule to not only exploit the dipole as a binding site, but to use the dipole to make sure the molecule has the correct orientation within that site, so that then other binding modes can be used, such as hydrogen bonding. So this is why knowledge of dipoles is critically important. We need to appreciate that dipoles can be used in this way. Although, of course, you would not need to know specifically how a dipole would enter into a binding site. You simply need to know that it does, and it can be used to make the orientation of the molecule correct in order to promote binding through other modes as well. Now, the last binding mode we're going to look at is an ion-dipole interaction. Now, you can think of this as similar to dipole-dipole interaction. So this is where we have, on one molecule, we have a permanent dipole, such as this molecule here, containing a carbonyl group. This has a permanent dipole, as indicated by this arrow. And we have a second group that has an ionic charge. So, for example, this group here, which is a deprotonated carboxylic acid group. So when this molecule with a dipole approaches this group, it is able to interact with the negatively charged group through the region that is positive, that has a slight positive charge. So through the region which contains the positive charge in the dipole. Now again, it doesn't matter whether the permanent dipole is on our incoming drug molecule or our binding site. And it is possible for this to bind using both positive or negative charges. So for example, if this functional group weren't a carboxylic acid, and it were positively charged amine instead, this amine could interact with the oxygen in the carbonyl, which holds a slight negative charge. Now what can also happen is that the charge on an incoming molecule can induce a dipole in another molecule. So for example, here we have a positively charged amine, and when this approaches close to an aromatic ring, it can induce a dipole in that ring. So where the electrons are attracted towards the center of the ring to interact with the positively charged amine, making the outside of the six-membered ring very slightly positively charged. And you can see that here. So we have the slight positive charge on the outside, the slight negative charge in the middle, and this slight negative charge is interacting with the positive charge present on this amine. Now, ion dipole interactions are not as strong as electrostatic interactions, but they can be used to drive that initial attraction of the molecule into the binding site. Because one of the atoms holds a formal charge, this interaction is felt over a larger distance. Now, not quite as far as an electrostatic interaction, but it can still be used in a similar way. So the two types of interaction that can be used to drive the molecule into the binding site are the electrostatic interaction and the ion dipole interaction. All of the others can only hold the molecule in place once that binding site has been entered. Now in the next section we're going to move on to look at protein structure. So we'll be looking at the primary, secondary, tertiary and quaternary structure of proteins, and we'll be having a look at how interactions between the functional groups in the amino acids can contribute to this structure.